So dear student, so today our topic is glycogenolysis. That is nothing but glycogen metabolisms. It is a process in which the glycogen is metabolized into glucose. My simple question is, so what is the glycogens? So anyone, if you know, what is glycogen? So why it is required to metabolize? Decreases. Okay, so when the plasma level of glucose decreases, the glycogenolysis is mainly occurs. That is the breakdown of glycogen and glucose is taken place in the muscles as well as in the liver, because liver and muscle, they are acts as the stores for the carbohydrates in the form of glycogen. So glycogen nothing but it is the one type of the biomolecule. It is acts as the EFD for the carbohydrates. So during the starvation, during the emergency, so then immediately the glycogen can undergo the glycogen metabolisms or glycogenolysis. Then it can release the, that is a glucose 1 phosphate molecule. Okay, so that glucose 1 phosphate molecule, that is essential for the synthesis of the glucose or the productions of the energy. Okay, so that's why the glycogenolysis is a process in which the glycogen molecule from the liver as well as in the muscles, it can break down into a glucose. In the case of emergency, it is helpful for to maintain the source of glucose or source of energy. So what are the glycogen? So that was stored in the liver. That glycogen is helpful for to maintain the source of the glucose in the blood. Okay, it can always, that was helpful for to normalize the blood sugar level. If the blood sugar is excess, then that glucose can contain to glycogen itself in the liver. Okay, so what were the glycogen that was stored in a muscle? It is acts as a source of energy, because muscles, so they are each and every our locomotions, we are depending upon our muscles. That is the contraction and relaxations of the skeletal muscle, each and every our daily activities. So that was, we are mainly depending upon the properties of the skeletal muscle, that is the contraction and relaxations. So glycogenolysis, the process is helpful for to rapidly to supply the energy. And the main features of glycogenolysis is it do not require oxygen. It can undergo the completely and aerobically, and also there is no need of energy expenditure. The every steps in the glycogenolysis, they do not require any kind of the energy. So these are energyless. So that's why in the case of emergency, the glycogenolysis, it can provide the source of energy, or it can provide the energy by continuously supplying the glucose. But regarding limitations, so the glycogen or glycogenolysis, it is the limited process, because the storage of glycogen is a limited. It has a particular limitations. So that much of glycogen only stored in the liver and muscle. So that's why, so it is a limited process, and also it can produce the low ATP, because so that glucose enter in the glycolysis, it can give the only around, that is the 2 ATP or 3 ATPs. Okay, so like that the glycogenolysis, it can also have one of the limitations regarding that it can produce the low ATP, as well as it is the limited process due to limit storage of the glycogen. And glycogen is mainly stored in the muscles as well as the liver and adipose tissues. So where they acts as the FD for the carbohydrates and FD for the glucose. So first we learn regarding the glycogen. Okay, so dear students, the glycogen is a polymer of glucose. It is acts as the primary carbohydrate storage in form in all animals. So in all the animals, including the human beings, so excess of glucose or carbohydrates, it is stored in the form of the glycogen. So that glycogen is mainly stored in the liver or mainly stored in the muscles. So during emergency, they are releases the glucose. And regarding the glycogen, so glycogen is the compositions of several units of glucose. Okay, glycogen is a compositions of several units of glucose. So those glucose are linked by alpha 1,4 glycosidase bond. Okay, so those glucose unit, so they are joined together by a one bond. So that bond was known as the alpha 1,4 glycoside bond, and as well as it consists of many branches. So those branches of the glucose residue or glucose unit, so they are bound together. So with the help of the alpha 1,6 glycoside bond. Okay, so these are the two bonds. So they are present in the glycogens. So glycogen, it is consist of the residue of glucose molecule. First, some glucose molecule around 8 to 12 residue. So they are grouped together. They have formed the one glycogen and they are interconnected with a alpha 1,4 glycoside bond, and as well as, so they are having the branches. You can easily you can see in the air. So they are having the multi branches, and these branches, they consist of the glucose unit, and those are bind by, that is the alpha 1,4 glycoside bond, and these branches, so they hold together in the unique. Okay, so that was by a bond, that is alpha 1,6 glycoside bond, and each glycogen molecule, it should be having at least 8 to 12 glucose residue, and target for glycogen is the breakdown of alpha 1,4 glycoside bond. So due to breakdown of alpha 1,4 glycoside bond, the glucose molecule was released from the glycogen molecule. Okay, so after that, certain glucose molecule, they are shift from this bond, from alpha 1,6 glycoside bond to the alpha 1,4 glycoside bond. The next molecule was ready for to release. So like that, in the glycogen, the targeting bond is a alpha 1,4 glycoside bond, and the alpha 1,6 glycoside bond molecule, they continuously replace the glucose molecule from there into the residue chain, that is the alpha 1 glycoside bond. And each glycogen molecule, so they are consist of the single that is the reducing and multiple non-reducing ends. So these reducing end, so they easily undergo the phosphorylations or phospholysis. Then the phosphate group is removed, then the bond is break, then one glucose molecule will be released into the circulation for glycolysis for many other process. So like that, the targeting in the glycogen is alpha 1,4 glycoside bond, and as well as non-reducing end of the glycogen, because glycogen, it consists of single reducing end and multiple non-reducing end. So regarding the glycogenolysis, so it is the process by which the glycogen, the primary carbohydrate stored in the liver and muscle cells, is broken down into a glucose. Okay, and to provide the immediate energy or to maintain the blood glucose level during the times of need in the case of emergency. So here nothing but the glycogenolysis is the process by which the glycogen can be metabolized into a glucose. Okay, or phospholysis of glycogen into a glucose. So that process was known as the glycogenolysis, and glycogenolysis, the process, it is helpful for continuously to supply the glucose, or helpful for to monitor normalize the blood sugar level in the case of emergency only. Okay, if the glycogenolysis is abnormal without need of the glucose in the blood, then glycogenolysis will be run or automatically it can occurs in our body, especially in the liver and muscle means then the blood sugar level is more than the normal. Okay, so that condition was known as the diabetes mellitus. If any problem in the glycogenolysis, if it is abnormally continue or it is occurs, it means so that was responsible for to cause of the one metabolic disorder. So that disorder is known as the diabetes mellitus. So next one, so regarding the glycogenolysis, so glycogenolysis is the process here the glycogen molecule can be break down to a glucose 1,6 phosphate and glycogen, that is n minus 1. N is equal to number of glucose molecule or residues in the glycogen. So in the next step, once the glycogen will be glycogenolized or metabolized, it can produce the one glucose 1 phosphate and glycogen residue is produced, but that will be the n minus 1, one glucose molecule will be the left from that glycogen. So that's why the glycogenolysis is the breakdown of glycogen to a glucose 1 phosphate and glycogen n minus 1. So glycogen, the branches, so they are catalyzed by sequential removal of the glucose monomer via a phosphorylasis by enzyme known as the glycogen phosphorylase. So glycogenolysis, it can occurs in the cytoplasm of the cells in the muscles like liver and as well as in the adipose tissues and as well as in the cytoplasm cells of the skeletal muscle. So where the excess of glucose stored in the glycogen. So during the emergency, so liver cells, skeletal muscle cells and adipose tissues, so they are undergoes the glycogenolysis or glycogenolysis can be taken place. So during the starvation, during the emergency when the blood sugar level is less than normal. So like that the glycogenolysis is mainly occurs in the cytoplasm cells in the skeletal muscle or in the liver and as well as in the adipose tissues. In the muscle, especially the glycogenolysis is occurs as a source of energy. Okay, in the muscles and the adipose tissues, especially the glycogenolysis was responsible continuously to supply the ATP for contraction and relaxations of the skeletal muscle. But whatever the glycogenolysis occurs in the liver, so that process is helpful for to maintain the sugar levels or it can provide the source of glucose only. So regarding the result, so the end of the glycogenolysis, that is the glucose 1 phosphate is produced from the non-reducing ends of glycogen chains. So that is the result of the glycogenolysis. Here one glucose 1 phosphate molecule will be released from the non-reducing ends of the glycogen chain. So this is the regarding the short introductions regarding the glycogenolysis. Next we learned process step by step. The glycogenolysis, it mainly consists of the only 3 steps. In first steps, the phosphate glucose 1 phosphate will be forms. In second step, the once again after losing of glucose 1 phosphate, the glycogen molecule will be rearranged and ready for another glycogenolysis process or another step 1. And step 3, so it can glucose 1 phosphate can convert into a glucose 6 phosphate. So regarding the step 1, that is the known as the release of glucose 1 phosphate from the glycogen. So in the presence of the or this reaction was regulated by the enzyme glycogen phosphorylase. So glycogen phosphorylase is an enzyme. So this was initial enzymes. So it is required for the glycogenolysis. So this glycogen phosphorylase, it can break down glycogen molecule at alpha 1,4 glycoside bound, okay, and produces the glucose 1 phosphate and glycogen, that is the N minus 1 residue. So here the two residues are there, here only one residue are left. So like that, and these glycogen, it will be having the residue or about, that is 8 to 12 residues are present. Step by step, one molecule can be releases or break down under influence of glycogen phosphorylase and by additions of the phosphate. Okay, so this reaction actually it is the phosphorylasis. It is not a hydrolysis. Okay, it is the phosphorylasis, not a hydrolysis, but this process is similar to the hydrolysis. In hydrolysis, the water can added. Okay, so due to additions of water, the hydrolysis is occurs. So due to hydrolysis, the larger molecule is counted into smaller molecules. But this step 1 process, it is not a hydrolysis, because here also the larger particle can convert into a smaller molecules or smaller particles or molecules. Okay, but it is a phosphorylasis because there is no water can be involved. Here only the additions of the phosphate group, the larger particle glycogen can convert into smaller molecule, that is glucose 1 phosphate and glycogen. So that's why it is differ from the hydrolysis, but it is similar to the hydrolysis. Only difference is instead of water, the phosphate group can be added. Okay, then process is same. Larger molecule can code it into smaller molecule in the presence of the enzyme glycogen phosphorylase, and this step it do not require any kind of the energy or ATP. So that's why the glycogenolysis it do not require any type of the energy, or it is less energy expenditure process. So regarding first one step, the release of glucose 1 phosphate from the glycogen. So later on in the step 2, the release, the continuations of step 1, that is the once again the glycogen phosphorylase only acts on the non-reducing ends of glycogen chain and are at least 4 glucose away from this branch. Okay, then after that it mainly break the, that is 1 4, that is alpha glycoside bond. Okay, in the step 1, and it is the phosphorylase. And step 2, that is the rearrangement of glycogen molecule. So this step 2 was regulated by the enzyme, that is glycogen debranching enzyme. It is also known as the GDE. So this GDE it can perform the 2 steps and involved in the rearrangement of glycogen, and as well as it can shift the glucose molecule from 1,6 branch into the 1,4 branch, because only 1,4 branch it can undergo the phosphorylasis, not a 1,6. So that's why, so this enzyme it can rearrange the glycogen molecule after first step, after phosphorylasis, then one glucose molecule from 1,6 glycoside bond or residue it transported to the 1,4 branch. So like that, the another molecule was ready for the to release or undergo for the first step in the glycogenolysis. So here this enzyme, that is the glycogen debranching enzyme, in the first step it can transport the rest of sugar monomer units from alpha 1 branch onto the adjacent 1,4 branch, leaving a single glucose at a 1,6 branch. Okay, so the leaving it transfer and one molecule glucose can be leave. In the second step, GD also catalyze the hydrolysis of remaining glucose. Okay, at the 1,6 branch point to release the glucose from the glycogen chain. So this GD, one side it can transport the monomer from 1,6 branch to the 1,4 branch. In the another side it can also regulate the hydrolysis. Okay, and release the one glucose molecule from the glycogen chain. So this is regarding the step 2. And step 3, that is the conversion of glucose 1 phosphate to glucose 6 phosphate. So these reactions was regulated by the enzyme phosphoglucomutase. Okay, so phosphoglucomutase, it is an another enzyme, that is the phosphoglucomutase. It is another enzyme. Our third enzyme comes under the glycogenolysis. It can convert glucose 1 phosphate into glucose 6 phosphate, okay, by a phosphorylations. So first the glucose 1,6 phosphate, that G1P, is converted into or phosphorylated into glucose 1,6 bisphosphate, that is glucose 1,6 phosphate. Here one phosphate group will be added, so that's why. So this is the first. Here in the sub step 1, in the step 3, it is also phosphorylations. And later on, so one more in the same enzyme, the phosphoglucomutase, then the one phosphate group can be removed, it can remaining only one. So then finally the glucose 6 phosphate or glucose 6 phosphate will be the form. Okay, so like that the step 3 is regulated in the presence of phosphoglucomutase, where the glucose 1,6 phosphate can converted into glucose 6 phosphate. So next one, so this is the regarding the 3 steps involved in the glycogenolysis. So this is the review regarding the glycogenolysis. So glycogen, so due to phosphorylations in the presence of glycogen phosphorylase enzyme, one glucose 6 phosphate forms, and second steps will be going on, that is the rearrangement of the glycogen in the presence of the GD enzyme. But here the glucose 1,6 phosphate, then it can convert into glucose 6 phosphate in the presence of the phosphoglucomutase enzyme. Okay, so then, so this glucose 6 phosphate in the liver and kidney, it can converted into glucose, that is the glucose 6 phosphatase. So like, so this glucose enter in our blood, then enter in the various cells or all type of cells, then undergo the glycolysis. In another side, so this like that the blood sugar level can be normalized in the case of the starvations or emergency by liver and kidney. So that's why the liver, the glycogenolysis in the liver, it was helpful for to maintain the sugar level, because of it convert glucose 6 phosphate in the presence of glucose 6 phosphate enzyme into glucose, that is helpful for to maintain the sugar level. But so glucose 6 phosphate in another side, it can directly undergo the enter into the glycolysis, then where by various steps it can converted into pyruvate, so in the presence of oxygen. But in the absence of oxygen, so that can convert into a lactate, so it is occurs in the skeletal muscles. Okay, skeletal muscles only the anaerobic glycolysis is occurs, but rest of the cell the aerobic glycolysis is occurs. And this pyruvate, it can enter into the matrix of the mitochondria. So where in the presence of pyruvate dehydrogenase enzyme, it can forms the acetyl CoA. Then acetyl CoA enter into a TCA cycle or citric acid cycle. Then it can responsible to form the carbon dioxide and water. Okay, so like that the glucose can regulate it. And another side, same glucose 6 phosphate, it can enter into the HMP pathway or pentose phosphate pathway. So where it can responsible for the formations of the 5-ribulose or 5-ribose sugar or ribose 5-phosphate, that is required for the synthesis of DNA and RNA. So like that the glycogen was responsible for to maintain the biochemical reactions that was related to or that was depending upon the glucose. Okay, so that was going to glucose 6 phosphate, then enter in directly going to glucose or enter in the TCA cycle or enter in the pentose phosphate pathway or HMP pathway. So this is the regarding the review of the glycogenolysis. How the glucose can be supplied continuously during the starvation by the process that was known as the glycogenolysis. So next, regarding the important enzymes, so those are involved in the regulations of the glycogenolysis. The first enzyme, glycogen phosphorylase, so it can break the, there is alpha 1 glycosidic linkage, or it can rebranch and then release the 1 glucose 6 phosphate. The main activity of glycogen phosphorylase is it can release the glucose 1 phosphate. If the enzyme activity decreases means the glycogenolysis decreases. If enzyme activity increases means the glycogenolysis increases. If the enzyme activity is overactive, okay, it is uncontrolled, then only the diabetes mellitus occurs. If any drug, of any molecule in diabetic patient, if it is inhibit the glycogen 1 phosphorylase enzyme means, so that drug is acts as a anti-diabetic drug, or that drug or that molecule was helpful for the treatment of the diabetes mellitus. Okay, so next enzyme, there is phosphoglucomutase. So this enzyme responsible for to convert the glucose 1 phosphate into glucose 6 phosphate. So like that, these are the two enzymes that was responsible for continuously supply the glucose from the glycogen in the case of emergency, or in the case of low blood sugar level or hypoglycemia. So regarding the certain molecule or component, they accelerate or stimulate the glycogenolysis. So those are glucagon from the liver. So glucagon always releases, okay, from the pancreatic gland and enter in the liver. It can stimulate the glycogenolysis. When sugar level is less, then only the glucagon is released, then enter in the liver and enhance the glycogenolysis. As well as epinephrine, it is released from the adrenal cortex in the case of stress, or in the case of the fight and flight mechanism. So where epinephrine releases and it enters the liver and muscles, and it can stimulate the glycogenolysis through a enhancing the cyclic AMP dependent protein kinase A enzyme cascade. So that enzyme was responsible for to phosphorylate or enhance the activity of the glycogen phosphorylase enzyme. So like that, the glucagon and epinephrine, they can accelerate the or enhance the glycogenolysis. So next one, the inhibitors regarding the glycogenolysis, like insulin, it is completely inhibit the glycogenolysis by inactivating this enzyme, that is the glycophosphorylase. So that's why the insulin is commonly used for the treatment of the type 1 diabetes mellitus or insulin dependent diabetes mellitus. If any abnormalities, then only we are suffering from the uncontrolled sugar level or hypoglycemia or hyperglycemia. So regarding glycogenolysis, it is continuously, it can supply the energy as well as it can helpful for to maintain the plasma sugar levels. So regarding significance, so the glycogenolysis is and play an important role in the fight and flight responses. So this is a response of sympathomimetic nervous system. So that sympathomimetic nervous system is helpful for in the case of emergency, continuously supplying the energy in the case of stress. Okay, it can protect the body, and failure of the sympathomimetic nervous system, the person can die within the 24 hours or within the 2 to 3 days, because so that system is play important continues to supply the glucose to our body by involving in the fight or flight response by releasing the adrenaline. So that adrenaline enter the liver and muscle cells, enhance the glycogenolysis. So that's why the glycogenolysis play an important role in the fight or flight responses. Okay, it also contribute the regulations of the glucose level in the blood. So those glycogenolysis which is occurs in the liver, so that was helpful for to regulate the or to normalize the blood glucose level, and as well as it is the helpful for metabolism of glycogen polymer. It is an important during the fasting or starvations, or when you are failure to take the meals, then but glucose content is required. So on that case, so this glycogenolysis is the beneficial. And also it continuously supply the energy to the muscle cells. So muscle cells always require the energy. Okay, it continuously supply the energy by providing the glucose 6 phosphate. Then glucose 6 phosphate enter the glycolysis. Further, it can enter in the TCA cycle. Then it can produce the ATP. So that ATP is required for the skeletal muscle contractions. Without ATP, the muscles are not contracted. If ATP is not produced in the muscle means then only we are suffering from the fatigue. Okay, if you are having a good ATP in your leg muscles, then there you do not have any fatigue. Even through the day your work. That is the role of the glycogenolysis. Then in hepatocytes, it is made the breakdown of the glycogen is for the release of glucose. Once again, it is helpful for the to maintain the blood glucose level or blood sugar and helpful for to the other cells. These are the significance of the glycogenolysis. Nothing but overall they are helpful for to supply the continuously glucose level to the other cells as well as so they are the source of energy or ATP.